Hello students, in a previous lecture we already discussed about sample collection, enrichment, isolation and classification of thermophiles. Now in this lecture we will study about adaptation and application of thermophiles. So in adaptation of thermophiles have a fascinating adaptations that allows them to thrive in high temperature environments. They have heat stable enzymes and proteins that can withstand the extreme temperatures. Their cell membranes have unique lipid compositions that provide stability at high temperature. Additionally, thermophiles often have a mechanism to repair DNA damage caused by heat. This adaptation helps them survive and carry out essential functions in their hot habitat conditions. In terms of molecular adaptations, thermophiles have unique genetic and protein structures that enable them to function at high temperatures. They possess heat stable enzymes with increased structural rigidity, allowing them to maintain their shape and activity in extreme heat. Additionally, thermophiles may have specialized DNA repair mechanisms to counteract the damage caused by high temperature. These molecular adaptations help thermophiles survive and thrive in their hot environment. Enzymes and other proteins of thermophiles often differ to some extent in their amino acid sequences from enzymes that catalyze the same reactions in mesophiles. In appear that a critical amino acid substitutions in only a few locations in the enzymes of thermophiles allows it to fold in way that makes them much more stable to heat, and as a result they function optimally at high temperature. Thermophiles typically possess lipid rich in saturated fatty acids in their cytoplasmic membrane, thus allowing the membrane to remain stable and functional at high temperatures. Saturated fatty acids from a stronger hydrophobic environment than do unsaturated fatty acids, such as hydrophobic environments helps for the membrane stability in thermophiles. Heat stability of proteins in hyperthermophiles is also improved as a result of an increased number of ionic bonds between the position and positive and negative charge positive and negative charges of various amino acids. This makes densely packed, highly hydrophobic interiors of the proteins which naturally resist unfolding of protein in the aqueous cytoplasm. Also, solutes diionisitol phosphate, diglycerol phosphate, and monosyl glycerate that are produced in high amount in the cytoplasms of certain hypothermophiles helps stabilize their proteins against thermal degradation. So next topic is application of thermophiles. The use of thermophiles for biodegradation of hydrocarbons with low water solubility is of interest, as solubility and bioavailability we enhanced at elevated temperature. The thermophilic bacteria have application in chemical feedstock and fuel production. Also, its use in bioconversion of waste. They use in enzyme technology, also useful in single cell protein production. Thermophilic enzymes have advantages for their uses in commercial application and particularly for the production of chiral compounds to produce optically pure pharmaceuticals. They can be used as a biocatalyst in the application of green chemistry. In biotechnology, thermophiles also used there. Thermophilic and hypothermophilic microorganisms offer some major advantages for industrial and biotechnological processes, many of which run more rapidly and efficiently at high temperatures. Extreme enzymes and molecular chaperones obtained from hypothermophiles are the examples. Extreme enzymes. The term extreme enzymes has been coined to describe enzymes that function at some environmental extreme, such as high temperatures and low pH. Since most of the industrial processes operate based at high temperatures, extreme enzymes from hyperthermophiles are widely used in both industry and research. And such extreme enzymes are proteases, amylases, glucose isomerase, polyolase, xylenases, glucoamylases, TAC, and PFU DNA polymerases, glutamate synthesis, and aminotransferases. So the proteases, that is a protein hydrolyzing enzyme that have been isolated, purified and characterized from Pyrococcus, Thermococcus, Sulfolobus and Staphylococcus and many other bacteria. A serine type proteases has temperature optima of 110 degrees Celsius and a half-life of 4 hours at 100 degrees Celsius. The serine protease from Sulfococcus mucosus exhibits its activity at 100 degrees Celsius. Then second one is amylases. Amylases have been extracted from Pyrococcus furiosus and Pyrococcus voiceus and are widely used in textile, confectionery, brewing, paper and alcohol industries. Then third one is a glucose isomerase. Glucose isomerase is widely used in the food industry. This enzyme converts glucose to fructose for use as a sweetener. It is used in food industry. Then next one is a pullulanase. Pullulanase second, that is a amylopullulanase obtained from the Pyrococcus ovis or a Pyrococcus furiosus, Thermococcus lithoralis or many other bacteria, has a temperature optimum, that is a 105 degrees Celsius and is useful for the bioconversion of starch into various useful products of industrial significance. Then fourth one is a TAC and PFU DNA polymerase, that is they are used in a molecular biology for the amplification of DNA using polymerase chain reaction, that is a PCR. TAC polymerase found in a Thermus aquaticus is active at 80 degrees Celsius at pH 8. Then next one is a glutamase synthetase. The glutamase synthetase, that is GS, is active at 100 degrees Celsius and is used for the synthesis of glutamine from glutamate and ammonia. So next topic we will study in next lecture. So thank you. Goodbye.